For the PCR method, we will need a primer, Taq polymerase, our DNA sample, DNTPS, PCR tubes, and a thermocycle. First, we will mix the master mix with our sample. The first 2-minute denaturation will open the strands of the DNA and it will ready to amplify 2 strands of DNA. In the annealing process, primers will align and attach, forming 2 strands, one forward and one reverse. In the extension, polymers will build the new strand. This process will take place 40 times. This will lead us to thousands of copies of the DNA with the markers that we want. At the end, we will keep them, all the copies, below 12 degrees and we will maintain them. Now we can do another procedure to discriminate our sample and decide if it is transgenic or not transgenic.
Six PCR tubes were set and labeled according to the corresponding content of each one, following the next table. There were 3 types of food used and 2 types of master mix used. The food was according to non-TGO or non-transgenic, test food, and TGO positive control. The master mix used were plant master mix with color green and TGO master mix colored with red. As the table shown, the 6 tubes were filled with 20 microliters of a type of food DNA and 20 microliters of a type of master mix. The solutions were formed using a fresh pipette each time and taking care of not transferring any instagene to the PCR reaction. The tubes were recapped and labeled and then placed in the PCR thermal cycle.
You can test the integrity of the polymerase chain reaction by identifying the photosystem II which is present on the plant DNA extracted from food. By using the PCR, you can identify the 35S promoter of the cauliflower multivirus and the terminator of the Nopalin simplase gene of Agrobacterium tumefaciens. If one of those sequences is present on the sample, it means that it's genetically modified organism.